In Chapter 4, we're going to be taking a look at the diversity of prokaryotic cells. And so if we look at this diagram, you can see the sort of tree of life and how we have the ancient ancestor. And the prokaryotic cells are thought to have evolved first, and these are the most diverse group of organisms on the planet here, with the archaea and the bacteria, the unicellular prokaryotic organisms that are unlike the eukaryotes over here, highlighted with the red lines, because the eukaryotes have the nucleus and other membrane-bound organelles that are not found when you're looking at prokaryotes. So for Chapter 4, we're going to be looking at the diversity of different prokaryotic cells. And there's a huge amount of information in Chapter 4 related to all these different groups that we see over here. And I'm working to try and minimize some of the information about all this stuff. And so really you want to take a look at focusing on this lecture and some of the stuff from the review to see what you should sort of focus your studies more. If you're interested in a lot of this stuff, there is, like I said, a lot of information in the book, and it is kind of interesting to get sort of the overall picture and to appreciate some of the diversity of the prokaryotic organisms that are out there. But if we look at this idea and think about the diversity in the microbial world, if you take a gram of soil, so think about just a scoop of soil like what you used in the microbe hunt lab, if you were to go in there and pull out the DNA and start to look at all the variety of different genes that are there, they estimate that using this method of looking at the DNA, they can find millions of different kinds of bacteria in just a scoop of soil out there. But when you look in Berge's manual, there are only a little more than 8,500 classified bacteria. And these are ones that have been cultured in the lab so that we can understand a little bit more about their metabolism and their ecological role when they're in the environment. So if you think about that discrepancy, million of bacteria in a gram of soil and 8,500 that have been classified really well out there. So if we look at this idea, a lot of these bacteria haven't been identified because they haven't been grown in the lab. We haven't been able to culture them. And a lot of them are going to need some special nutrients. And an example of this is if you look at some cells that are out there, they can be part of a very complex food chain. And so if you think about it, you have the first bacteria that eats some of the surrounding stuff in the environment, and it produces a waste product that's food for another bacteria. Well, if you think about that process, if we make a pure culture and we have the two bacteria in there, the one that makes its own sort of food is going to be able to survive. But the other ones that are out there, they need that first one around. And by process of making a pure culture, you've gotten rid of the first one, which means you have to be able to find the food that is appropriate for the second one. And so that can make it really difficult to make pure cultures, which is how we like to grow these things. Remember that idea when we look at microbes in the environment, they're growing as a community and they are interacting with a lot of other organisms, both microbes to multicellular things like plants and animals. But like I said, for this chapter, we want to focus on our prokaryotic cells. And remember, when we look at prokaryotic cells, they're found everywhere. They are all over the place on the planet. All they really need is carbon, energy, and some water available. And we can find them growing on top of mountains and at the bottom of the ocean. And you can also find them even in something like glaciers that are out there. When we look in the environment for prokaryotic cells, they are extremely important because they are involved in recycling elements that are out there. So when we look at photosynthesis, we always think about plants, but we don't consider the algae that are out there. We also don't consider the cyanobacteria, which are everywhere on the planet, and they are also doing photosynthesis, and they can use that light energy and produce as a waste product oxygen gas. So obviously we need that for ourselves. They're also going to be involved in recycling things like nitrogen. They can recycle phosphorus and get access to it in the soil, and also something like sulfur. So these elements that are out there are going to be recycled by the soil microbes as they're breaking things down and releasing them from something like rocks or plant roots that are out there. Likewise, because they are involved in this process of recycling, when we look at our prokaryotic cells, they're decomposers. So they're going to take dead plants and animals and other organisms and break them down to their components. And so effectively they are refertilizing the soil and are involved in this process of recycling some of these elements that are out there to make them available again for the next generation of organisms. And unfortunately for us, some prokaryotic cells are human pathogens and are going to cause diseases. And when we look at this, it's kind of interesting that 5 pounds of you is estimated to be something else. So microbes that are growing on and in your skin, and also they are inside of you right now in your GI tract. And some of the pathogens are things that can eat through your skin, or if they get out of your gastrointestinal tract, now they're getting access to your body cavities where they can cause a variety of different kinds of diseases and cause harm to you as they decompose you, and they are literally eating you from the inside out. When we do look at prokaryotic cells, another distinctive feature here is prokaryotic cells are not able to do sexual reproduction. They are going to be able to reproduce through an asexual process that's known as binary fission, and it's summarized with this figure from the text. It's pretty easy. When you look at it, the DNA is going to be replicated. So remember, prokaryotic cells, they've got circular chromosomes that are actually attached to the cell membrane somewhere in there. So here the cell has duplicated its circular chromosome, and then the cell is going to get a little bit bigger, and it divides in half right here, forming that cell wall or septum between these two cells, and then your daughter cells are going to separate. And here you can see two bacillus cells, or bacilli for plural, that are going through this process of cell division, and there is your cell wall or the septum that is forming between those. When we look at this process of binary fission, this is a general sort of diagram, but we can see some different examples of this. And here is just a cartoon showing you what's known as a budding cell. So as opposed to here, where you've got the cells being equal in terms of their size during division, here you actually have a part of the cell that is budding off of there. So the nucleoid region, or the DNA with the chromosome there, is going to be in these two parts of the cell, and this one just breaks off and it's not equal to the parent cell that is there. Sometimes with these kinds of prokaryotic cells that come in like this string that you can see right here, those strings are going to fragment. So you're just breaking off portions of this after the cell has gone through that binary fission. And some kinds of gram-positive cells that are out there are going to do an interesting type of binary fission that's known as snapping cell division. And here they actually are going to deposit parts of the inner cell wall across the dividing cell there, and eventually they're going to build up enough tension here that it will almost snap the two together. And so you get these two cells that are sort of off at an angle together, and they're still connected at the beginning of this process. So all of these can be ways that we can start to use for classifying our prokaryotic cells. If they do this process, it's different from the other cells. So remember that idea we looked at the beginning in chapter 1 with taxonomy, that we have different categories that are available to classify the cells. And another really unusual form of reproduction that we find in prokaryotic cells is something that is found with a bacteria known as Epilopicium. And this bacteria is found in the stomach and intestines of sturgeon fish, which people say, why would you want to look at sturgeon fish? Well, that's where you get caviar. And if your sturgeon fish are dying off, people are taking a look at what's going on in the food to see if maybe that's causing these caviar-producing fish to die off, because there's a huge amount of money that's made off of caviar, so you want to have those fish. And they identified this weird kind of thing you can see in a differential interference contrast photomicrograph. So if you remember, the Dick microscope is able to use two types of light that are in phase to give you really, really high contrast. And when they looked at these cells, they saw this weird kind of thing inside of it, and for lack of a better name, they initially called them spores inside of the cells. And one cell can form between 1 to 12 of these spore-like things inside of there. But it turns out this is actually the daughter cell that is inside of there. So this one cell is dividing internally over and over and over to produce this string of other cells inside of there, and then those live offspring are actually going to pop out of the body of the mother cell there. And this is something that is known as viviparity. So it's just kind of a weird thing to think about, that you've got all these babies inside the mom, and then they burst out and come shooting out of the belly of that dead cell that was there originally. When we look, just to sort of get you to think about things, there is a project that is being carried out using your tax dollars known as the Human Microbiome Project. And what they're doing is they're actually starting out, and they've characterized communities of microbes that are found at 5 different sites on the human body. They looked in the nasal passages, the oral cavity, your skin, your GI tract, and also the urogenital tract. And initially, they started out looking at ribosomal RNA genes. And remember, ribosomal RNA genes are going to be involved in the process of making the ribosomes. And all cells that are out there need to have ribosomes because those are the protein factories. So by comparing the sequence of these genes that are found in all the cells, regardless of if you're prokaryotic or eukaryotic, we can start to place these organisms into these different groups. You look for conserved sequences and say, these guys all have the same thing, so we're going to classify them together. And it's really kind of an interesting project there because they've actually been able to move on to a second phase. Not only are they looking at the population of these different groups or communities of organisms, but now they are actually starting to find and correlate that the organisms that are present in people are different from what we find in people that have different kinds of diseases that are out there. And so if you look at this, we can look at different locations in the body, and here we can see a variety of different ones that are being looked at with the intestinal tract. And so you can take your baseline, what you would find in the normal healthy person that was out there, and then start to compare that with a person who has, in this case, inflammatory bowel disease. And you can look at the different groups of bacteria there and notice there is a distinctly different profile of organisms that are in this patient. And so more and more we're starting to learn that not only are you made of what you eat, but also the things that are in your intestines and what they're eating and what they're producing. And by shifting the population of organisms in a person, that can actually produce different types of diseases. And the hope is maybe we can shift the population back to the healthy control and alleviate some of what's going on with some of these diseases. And notice type 2 diabetes. There is actually a correlation showing that some shifts in the community of organisms in your intestines can actually aggravate the condition of diabetes, where you're not able to process your sugars efficiently. And so we'll be taking a look at some more of these things. And when we look at communities of organisms, we can find what's known as a symbiosis. And when you look at a symbiotic relationship, it's basically a interaction that is typically long-term between 2 different biological species that are out there. And I stole this figure from a Wikipedia paper that was out there because it shows you some of these different versions of symbiotic relationships. If we look at a mutualistic symbiotic relationship, here you've got a long-term relationship between individuals that are of different species, but notice both species can benefit from this process. So you've got a positive for species A and a positive for species B. A good example of this for the microbes is the bacteria living in your intestines right now. They're helping to digest your food, and they're also giving you vitamins. You are giving them a nice, warm, safe environment with this near constant supply of partially digested food for them to be able to eat. So both species are benefiting from this relationship there. When we look at commensalism, here it's different. One organism is benefiting from this long-term relationship, but the other one is kind of like, they don't really care one way or the other. And if we look at commensalism, microbes that are on your skin, they're eating your food and your secretions from your body, and they aren't really doing anything to you. Or microbes that are out there in the soil. These kinds of things They can benefit from another organism that is producing different forms of nitrogen, and that can definitely benefit the plants that are out there because they'll be getting fertilized with these nitrates, but other organisms are just kind of living in the surrounding area. If you've got a parasitic relationship here, the parasite often is going to live on or in the other organism, the host, and that will cause harm to the host. So the parasite benefits and the host is harmed. So kind of an interesting thing to think about. A really good example for this would be tapeworms. They're getting bigger because they are basically absorbing your digested food, and you're getting harmed because you're losing access to that food. Some of the other relationships that we can see, neutralism, as the name would suggest, both of the organisms are neutral. And there are arguments about this, that how can you have two species that are living in a long-term relationship where they don't affect each other? And the best example for this would be something like spores. Spores are dormant, so they don't have any effect on other cells, and other cells aren't going to have an effect on them. But like I said, there is argument about this symbiotic relationship that's neutral. The last one that we can really see here is what's known as amensalism. And this is kind of a weird one because here you have one organism that is harmed while the other one is unaffected. And so if you think about the microbes that are on your skin, like staph epidermidis or Prokaryotic bacteria that causes acne that is out there, these organisms have the potential to cause diseases, but they do also produce chemicals that are going to kill off other cells that are out there. So the organisms that are living on your skin are going to be unaffected, but the other organism that is living on your skin could be harmed by the presence of these chemicals that are out there. So think about these different kinds of relationships between organisms when they're living together, and how sometimes it'll help, sometimes it'll hurt, and sometimes it's not going to have any effect on you.